Okay, welcome back to the second video here. And I want to get into moving a little bit beyond vicariance biogeography, and I'll explain what I mean by that here in a second. Basically, what I just mentioned in that previous video was getting at how there's this assumption when researchers are usually looking at these things, there's this assumption that vicariance is what happened. And even though that may be incorrect, the initial assumption is usually vicariance when you're looking at how or why organisms may have diverged. And that's because, as we saw, dispersal has so many other ways. It's a lot harder to disprove dispersal than it is vicariance. If you have any indication that dispersal happened, then you can immediately discard vicariance. So it's a good starting spot to at least advance your understanding of that. But we sort of started to move beyond that approach as well as we have more and more tools and more data. And so let's sort of look at that. And we gotta have to go back to this idea of the taxon area cladogram first. And I want to introduce sort of this idea of congruence, which is, if we have our taxon cladograms, and maybe we have cladograms for several different types of organisms here. This is all hypothetical, so it doesn't really matter what they are. But let's say you have, groups 1, 2, and 3, which might be similarly related, and 4, 5, and 6, and 7, 8, and 9, all occurring in the same area, but they have different taxon cladograms. And so if you construct taxon area cladograms for all of them, you can sort of overlay them either visually or statistically and get an idea of how much in agreement they are. Are things diverging at the same time or not? And this is going to give you an indication of whether vicariance or dispersal is at play here. And the degree to which that these line up with one another is something called congruence. If they line up perfectly, then you have perfect congruence. And as we have seen over and over again, and hopefully you're getting the sense for, is that almost never happens. Things are really complicated when you're looking at the real world, like the physical world, and how organisms move around, and evolution, and things are messy. So you're almost never going to get perfect congruence, and also because the data we have isn't perfect. But it's a tool that you can use to get some level of confidence and get an idea of whether your hypotheses about how these things occurred are correct. So I think it makes a little bit more sense though if we sort of break this down to step by step. So this is from a study published in 2009, and basically proposing like a fundamental approach to doing these kind of studies. And this is like the 5 steps in an evolutionary biogeographic analysis. And so the numbers indicate the sort of the steps. So you're going to start with the biotic components, and maybe you're inputting distributional data. Maybe you have pan biogeography data in areas of endemism, and you might have phylogenetic data as well. You combine the biotic components with the phylogenetic data, and you can get to what we've been looking at, which is pretty much cladistic biogeography. And then that can help you corroborate some of the biotic components, and you can get some regionalization ideas about how organisms in this region might have evolved. And then you can sort of go beyond that and say get some xenochrons, and this is just a term basically which corresponds to like groups of organisms that have similar evolutionary descent. And you can use molecular data at that point, paleontological data, and those things like molecular clocks that we looked at, and combine that with geological data. And ultimately, you can get some what they're calling geobiotic scenarios, which is basically, for large groups of organisms, you can get really high detailed evolutionary histories and lineages and look at how things diverged over both large scale time and space and across lots of different taxonomic groups. This is just sort of to show like how far the analysis has come here. So what we're looking at here is the historical biogeographic processes incorporated into an R package, which is Biogeobears, which shows the range of modern approaches and scenarios. So if you don't know what R is, it's a programming language that's very frequently used by scientists. It's free, it's open source. I actually use it all the time. But it's basically a statistical and programming language for doing data analysis. And without getting like too much into the weeds on this, basically, we've moved beyond just taking into account sort of basic vicariance or dispersal events. There's all sorts of events that can be teased out if you have this sort of distributional data, as well as like phylogenetic data and maybe some of the climate and relationships to that. So we can look at dispersal and extinction, movements in ranges, sympatric speciation, vicariance, and founder events, and all sorts of things can be put into these models, and you can create models that will help explain all sorts of different scenarios, moving beyond just simple sort of vicariance. So I'm not expecting you to know all of this means, but just to get the sense that modern techniques are moving beyond, well beyond, originally just center dispersal theory, things like that. And just to show you an example of how powerful some of this analysis can be, here we're looking at the phylogenetic histories generated from that R package, Biogeobears. So the geographic range, evolution, and phylogenetic structure of the plant genus Salvia, which is here in the Western United States. So, we can reconstruct the phylogenetic structures here and get a time for when things diverge from one another. And obviously that's really powerful and important information to have and really useful and valuable. But more than that, what you're seeing here in the node, like the color of these individual nodes, is giving an indication of the probability of where those ancestors, where current species and where past ancestors were likely to have existed. So you can really reconstruct not just the phylogenetic structure, but the geographic range of those ancestors and how those have changed over time, and get a really much more in-depth picture of the evolutionary history of this groups. And so you can sort of see like how powerful that can be. Like, oh, we can not just look at the, how these organisms are related to one another. We can also look at where they might have existed and how that might have changed over time. So some pretty cool stuff. Okay, so I want to look at a few more examples here, and I want to come back to this idea that I've sort of introduced, but it's really the crux of what's going on here, and that is that even when geological history is simple, evolutionary histories can be complex. And so even if the geologic history isn't that complex, the understanding the evolutionary histories can be difficult because organisms can move around a lot. And Hawaii is a great example of this. So if we were to just look at the area cladogram that's predicted for the modern large islands of Hawaii, we can see just a simple progression rule that takes into account the age of the islands. And it's really simple. You have the oldest island here, and then the other islands are younger and formed, probably in this sequence where you have these islands more closely related to one another geologically in time than these two, because these are older. So basically this one formed, and then this one formed, and this one formed, and this one. It's very simple, nothing complex really going on there. And you might expect, if vicariance is what's driving most things, you would expect that you would see similar sort of taxon area cladograms for organisms that are found on Hawaii, right? If an organism showed up here, and then it may be dispersed over here, or something like that, where you have this progression rule, we should see similar sort of patterns in the phylogenetic cladograms of these species. But what we can see, so okay, so let's just look at a few. So many Hawaiian fauna groups arose from a single ancestral colonization event of the island. So that would make sense. You would expect to see a single colonization event and then maybe diversification as the individual islands showed up. And here we're looking at the mitochondrial DNA phylogeny for the Hawaiian vinegar flies. And we do see a single colonization event here, this ancestor here. There's a lot going on here. I'm not going to get into all of this. But you see this single colonization event followed by diversification throughout the islands by this group, and that's more or less what you would expect. Something interesting that happened here is that so you have this group of flies, a separate lineage formed. So basically, you had this ancestor that showed up in Hawaii. You had diversification along this lineage, but then you had a second lineage that formed and then left Hawaii and actually became very, very widespread globally. So kind of an interesting thing where an ancestor shows up, evolves, an entire lineage evolves from that ancestor and then leaves and goes back out into the world globally and disperses. So kind of interesting, but that's kind of a side note and sort of unrelated to the point here. Mainly like this would fit probably the model where you would expect, and you expect to see some lineages that are older than others, which you can get a sense of for how shallow or deep a deeper sort of longer lines here are going to basically indicate older lineages. So this is more or less what you would expect to see. It gets a little bit trickier, like some of these when you start looking at specific colonization events. So this is a mitochondrial DNA phylogeny for the Hawaiian tree snails supporting a single colonization of the island of Oahu. And so it's a little bit different. I mean, the progression rule was like this was the oldest island and then sort of progressively younger as we moved in this direction. But here, the snail there was an ancestor that arrived in Oahu originally, and then you're seeing those dispersal events and diversification across that. So it fits a little bit, but slightly different. However, so some of those fit what we would expect to see, but some other groups such as orb weaver spiders and land snails looked like they colonized the island more than one time actually. So if we look at the molecular phylogeny for the Hawaiian amber snails, which supports multiple colonization patterns and events. So basically, you have two clades here, and each clade indicates a separate colonization event of the islands. And so I think clade A is in the solid line and clade B is in the dashed line, I think. And so basically you have two separate events which have produced two separate distinct lineages within the islands, within these groups. And so you can see it gets really tricky. Even if you have this relatively straightforward geologic history of these islands, the taxonomic and evolutionary history of the organisms found there don't necessarily 100% correspond to that. And basically it comes down to dispersal. Something we haven't even taken into account really, so this is basically all just looking at allopatric speciation, where you have some barrier, either organisms disperse over a barrier or a barrier forms, and you have speciation occurring. If you remember when we talked about that, speciation occurring between those two populations. That's not the only type of speciation we can have. In fact, we have evidence that there might be sympatric speciation going on for the Orson-Welles spiders, where you basically have divergences that happen within these islands, and you have these groups diversifying from one another within the islands. So basically you have this sympatric speciation, which sort of just adds another layer onto it if you're trying to reconstruct the evolutionary histories using the geologic histories. You have stuff that doesn't correspond to the geologic histories at all. So you could have dispersal, but you could have no dispersal and still have speciation. So it gets really complicated pretty quickly when you're trying to tease these things out. Okay, so I want to sort of finish up by reinforcing this idea of congruence and hopefully illustrating that a little bit more. So what you're seeing here in this diagram is two lineages, like these different hypotheses or different scenarios of taxon area cladograms. So you have two lineages that exist for this group 1 and 2, both of them occurring in areas A, B, and C. A, B, and C, A, B, and C. And if you have congruence, if you have total congruence, that basically means that across space and time here, things are lining up across space and time. So we can see the splitting is occurring at the same time. This is our time axis over here. And we have the same, and it's following basically the same area splits and taxonomic splits. So the lineages of these two are congruent in time and space. Pseudo-congruence, it would be where things do not line up in time. So they line up in space. You have the same basically area cladogram here, but taxonomically things are not splitting. If this is our time axis, you can sort of see how it's not, like this split is down here in this group, in this group it's up here, in this group it's down here. And so these splittings are not occurring at the same time. So that would be pseudo-congruence. And basically that would indicate that it must not be vicariance that's driving this. It's not the separation and the creation of a barrier is probably separate dispersal events. You can have incongruence, basically where you have things lining up in time, but the specific splitting is occurring differently. So basically these organisms diverge differently from one another in different places, and the splitting doesn't really line up there. Or you have pseudo incongruence where things basically just don't line up at all. They don't line up spatially. You have different taxon area cladograms, and the splitting occurs at different times. So this this would be one of you look at that and say easily, oh, this is just, they're not really, different scenarios are creating these lineages, and basically any of these would indicate that it's probably not vicariance that's driving the evolutionary histories of the organisms that you're looking at. So it's a little bit tricky, but I think if you spend some time with it, you can wrap your head around what's going on here. Basically, you're just looking at do the splits of these lineages occur? Do they line up over space and over time? And that can indicate that, oh, it was probably a vicariance event. If they don't, then it was probably a dispersal event. Okay, so yeah, I'm gonna finish up here this video. Keep working your way through the module. There's a reading about a Hawaiian flower species, so take some time and read that and work on the questions, and I will see you in the next video.